Hello, everyone. Thank you for joining our very first online Immunology Journal Club. We're going to start this journal club with a 30-minute presentation by our scientist. Today's speaker is Patrick Brouwer. He's our senior scientist. He used to work with PSC-derived T-cell development, but he's now joined STEMCELL, where he's working on our lymphocyte differentiation protocols. Patrick has joined us to talk about human in vitro T-cell development. And all I have to say is thank you, Patrick, for joining, and I'm going to hand it over to you. Thank you.
So good morning, everyone, or good afternoon, depending where you're living or listening from. So I want to start off with a disclaimer that the publications are open access and that STEMCELL did not have any part in the actual work. So this is the author's work that I'm presenting. So as Maria mentioned, I'll be talking about human in vitro T-cell development. I'll give you an overview and also some applications. So a brief outline, I'll start with the background as mentioned. I'll talk about disease modeling, different in vitro systems, and also get into some cancer immunotherapy applications. So some of the potential applications of T-cells that could be relevant in the clinic are disease modeling and therapeutic development or validation. And the Temeli 2020 paper actually talks about that mostly. There's also an application where you could grow normal T-cells or T-cell precursors, and these could be clinically relevant or useful for cases of lymphopenia, such as primary immunodeficiencies or acquired immunodeficiency syndromes, or depletion of the immune cells due to surgery or aging. Pretty much once you hit puberty, things go downhill from there. So your time is actually involutes and you're getting fewer and fewer T-cells as you age. Another application would be to generate antigen-specific regulatory T-cells. These would be useful to prevent tissue damage due to autoimmunity or due to organ or tissue transplantation. Another kind of aspect of antigen-specific T-cells are the effector T-cells. These would be useful for cancer immunotherapy. You could expand tumor-infiltrating lymphocytes or TILs, and you can even engineer your TCR with specific specificity, or CAR T-cells, of course, are another aspect. And the paper by Guo from 2019 kind of talks about in vitro methods, really changing the method that you can use, as well as getting into some of the cancer immunotherapy aspect of it. And Nagano 2020 paper, they're really more specific about making some cancer-specific T-cells for therapy. So first, a brief overview of how in vitro T-cell development works. HSCs start in the bone marrow. They generate lymphoid progenitors, which then migrate which can migrate to the thymus, where T-cell differentiation continues. In the thymus, they start as double negative DNs. Double negative refers to CD4, CD8 expression. They then mature to the next stage, which is immature signal positives that express CD4 in the case of humans or CD8 in the case of mouse. They then express the other one, so they're CD4, CD8 double positives. And they also undergo positive negative selection in the thymus. 98% of the cells actually die, they don't make it. But 2% that make it through become CD4 or CD8 signal positives that can get into the periphery. Kind of a step back developmentally speaking, pluripotent stem cells can differentiate into 3 germ layers. One is the ectoderm, or outer layer, endoderm, the inner layer, or the mesoderm, which is the middle layer. The mesoderm specifically can go towards the hemogenic endothelial stage and further differentiate to the progenitor cell stage. And this can give rise to then different kinds of blood cells that you see here. Another sort of version of this is starting with hematopoietic stem cells, such as, for example, cord blood HSPCs or hematopoietic stem progenitor cells. They can also give rise to progenitor cells. This would be more in the adult case, of course, not development. And same, they would give rise to different kinds of blood cells that you need for the immune system. Now, different kinds of in vitro T cell development, developmental approaches. These are sort of the two classical ones that I kind of used to outline in vitro methods. So the first original method to grow T cells in vitro was the fetal thymic organ culture, or FTOC. And basically what you do is you take a embryonic day 14 thymic lobe, you extract from the mouse, and you then conceive these with your progenitor cells of choice, really, and differentiate these then in the thymic lobe towards T cells. And this uses an air-liquid interface. There's different versions now based on kind of this method. Also, in 2002, the Syndicate Lab, which I was part of at one point, prior to STEMCELL, they really spearheaded a cell monolayer system that basically replaced the fetal thymic organ culture. And that's using a OP9 mouse bone marrow stromal cell line that expresses a notch ligand, delta-like 1 originally, or now delta-like 4 or others. And this really enabled T cell development in vitro in the absence of a thymic organ. For mouse PSCs, it's a little bit more straightforward. You can actually culture these on OP9s or stromal cells right away to get progenitor cells, and then you go on to this OP9 expressing delta-like 1 or 4 notch ligand to further allow T cell differentiation to occur. However, for the mouse, it's simpler. Humans, a different story. You need to first subject these to a pre-differentiation step. And in 2012, the Keller Lab, in combination with the Syndicate Lab, pioneered this sort of embryo body differentiation that allowed the generation of progenitor cells. And once these are seeded onto the OP9 stroma with a notch ligand, allowed for T cell development. This brings me to the first paper, which deals with the PSCs and their use for disease modeling. So why would you need to use PSCs? Often for, just an example, for primary immunodeficiencies, the patient tissue is just not available. Most models do exist, but they often do not really recapitulate the human disease the way it would be in the human, of course. And there's also some developmental differences between mouse and human T cells throughout T cell development, such as the DNA stages they have different markers, and as I mentioned briefly before, that they go through a different immature single positive stage. Now, using human PSCs circumvents all of these issues. Which brings me to the first paper, as I mentioned, is by Maria Temele from 2020. Using the RAC2 gene to model severe combined immunodeficiencies, or SCID. So first, at the top here is RAC2. It consists of a core element with a hinge and a plant homology domain. And this protein is essential as part of a complex to recombine the T cell or B cell receptor locus. Now, in this case of SCID, there's a point mutation causing a early stop codon, and so then truncation of the protein, and there's basically no function left. So what this looks like in a wild type person, the B cell development, you see the pro B cell stage here, and the pre B cell stage here, that's IgM negative, as well as the cytoplasmic IgM positive pre B2 stage. And then you have the immature B cells that are IgM positive at the surface. For the RAC2 SCID, so the truncated RAC2, you do see a similar proportion of pro B cells. However, the remaining proportion of B cells is all pre-B1 stage. So there's no cytoplasmic IgM at any of the stages that's present in the wild type. This is what the RAC2 SCID mutation looks like. It's a single nucleotide substitution that causes early truncation. Now, the group also gene repaired this, restoring it back to the right nucleotide and restoring the arginine back and the full protein. So just to show you what this looks like at the early stage, at the top is a wild type H1 that they used as a control. Middle panel is the RAC2 skid, so the RAC deficient iPS cells that they generated, and the gene repaired or gene corrected RAC2, which is also wild type RAC2 function. So at the early stages, there's really no difference in terms of hematopoiesis. You can see that there's a CD34, CD31 endothelial, hemogenic endothelial cells. And as well at this time point, day 12 is also 43 and 45 expression, so hematopoietic, early hematopoietic lineage. Looking later, in terms of T cell development, you see that there's a sort of a T lineage marker, CD7 is expressed. It's positive in each of these cases. There's also CD5 expressed. That's sort of the next T cell marker that comes up, normal for the wild type H1s, as well as for the RAC2 gene corrected. However, the RAC2 skid, you do see some CD5 expression, but it's highly reduced compared to the wild type counterparts. And looking at CD4 ISPs, immature signal positives, again, you see a great reduction at, for this stage, as well as the double positive stage, you see a great reduction compared to wild type, which has lots of CD4 and CD8 double positives. Now looking at rearrangement, this is what a normal rearrangement would look like for the T cell receptor locus, looking at this, these VJ segments. Of course, this is completely absent in the absence of recombination, as you can see for the RAC2 skid. And for the gene corrected, you see a fairly normal recombination occurring as well. Now, they went a step further to look at other cell types that they observed in a co-culture. So see the CD7, CD56 sort of NK cells, they don't see really a difference in that population for the wild type versus the RAC2 skid. However, they did observe that there's a CD7 negative, CD56 positive cell population, and that's enriched in the skid, while it's not really that highly present in the wild type or the gene corrected. And this is what the plot looks like, CD56 with CD7. And further looking at this population, they looked at different NK cell markers. They did see the presence of 33, which is not normally expressed in an NK cell. They do see very little, basically no CD16 expression for these NK-like cells, nor CD14 expression, which is just kind of a negative control since it's not expressed in NK cells either. However, what would be expressed in an NK cell would be NKG2D, and that's absent for these NK-like cells. So they summarized their findings by showing up different blocks, different stage blocks from the early thymic progenitors, the C7 positive stage, as well as the partial block from the C7 to the C7 and 5 double positive stage, and then pretty much complete block prior to the ISP stage. And they also observed these NK-like cells that are CD7 negative, but CD56 and CD33 positive. So that brings me to the next section that I want to talk about, and that is transcription factor mediated differentiation. So as an example and as a contrast at the top here, you can have a differentiated cell. And in 2006, Shinya Yamanaka basically made world news by using reprogramming factors or transcription factors to reprogram these cells that are differentiated into an induced pluripotent stem cell state. Now, almost looking at it from the other way, after that, some groups have also tried to use pluripotent stem cells or progenitor cells, and then use transcription factors to basically drive or force differentiation of the lineage of their choice. And that's with the use of lineage-specific transcription factors. So that brings me to the second paper, which is by Guo from 2019, November. And they try to use these transcription factors to drive T-cell differentiation from PSCs. But I want to point out that they use mouse embryonic stem cells. So this is their initial sort of protocol that they use. The first stage takes 11 days, and it's the induced hemogenic endothelial cell induction phase. At first, they have just a pre-differentiation And then they have a ESC line that has inducible RUNX1 expression, as well as different transcription factors that they tested that's in place of this X here. At day 6, they induced RUNX1 with one of the other transcription factors, and also in combination with different cytokines, as you can see here. And day 11 onward, they then enter the induced hematopoietic progenitor cell maturation stage, and they still keep the expression of RUNX1 with the other transcription factor on. At day 21, then they have they start the induced T cell functional assessment. So that can be done in the mouse by injecting the HPCs, induced HPCs. And 48 weeks later, they look at T cell emergence, and you can see by CD3 staining here. And just as a repeat of different experiments, you can see a very robust expression of CD3 induced T cells in the peripheral blood. Characterization of these induced T cells looks like this at 5 weeks. They looked at spleens, lymph nodes, and peripheral blood for TCR alpha beta expression. And you can see a robust expression of TCR alpha here, TCR beta here. And these are either CD4 or CD8 signal positives, as you can see in each of these compartments. Now, looking at the heat map here, they compared CD4 regular T cells with CD4 induced T cells, and you can see that by these gene expression of surface markers, virtually identical expression pattern, as well as CD8 versus induced CD8. Again, virtually same gene expression pattern of these surface markers. And they included analysis of B cells and myeloid cells as contrast, or negative kind of negative control. Here at the bottom is the heat map of genes that are encoding transcription factors. And again, you can see a very identical gene expression pattern for the CD4s versus induced CD4s. And the same is, again, the case for CD8 and induced CD8s. And again, B cells, myeloid as a contrast. They also looked at TCR receptor diversity, looking at thymus and the spleen, looking at a TCR alpha chain, and you can see a diverse rearrangement combination. As you can see by the different colors and different combinations, as well as for the TCR beta chain, as you can see in the thymus and in the spleen again, different diversities. These experiments led them to this sort of modified protocol, and they really want to test a scenario where they have induced T cells with a specific TCR of their choice, and they want to use this to suppress solid tumor growth. So how they go about this is using MEFs, or really a somatic cell, reprogram these using Yamanaka factors to generate iPSCs, or induced pluripotent stem cells. These are then gene edited with the inducible transcription factors, RUNX1 and HOXA9, which is the transcription factor that they found to be the best for T cell differentiation, or T hematopoiesis. This then generates the induced R9 or IR9 iPSC line. They further gene edit this to express their OT1 TCR, which is the alpha chain and the beta chain together, and they also include GFP in this cassette. And this is then the OT1 IR9 iPSC. They then differentiate these in vitro to the induced hemogenic endothelial cell stage, and further to the induced hematopoietic progenitor stage. They inject these then into mice along with the EG7OS tumor cell line. So just as a control, they're checking their OT1 IR9 iPSCs for expression of their TCR alpha and beta, which they've done here. This is intercellular staining at this stage because they're just testing the iPSC stage. So the T cell receptor would not be at the surface, but intercellularly it is expressed since it's a ectopic gene expression. And further, they looked at different mice for the presence of their TCR alpha beta chains that they ectopically inserted into the iPSCs. And in each of these 3 mice, you can see expression of TCR alpha beta, which is visualized by GFP expression. And these are then expressing the TCR alpha and beta of their choice. And that's the case for all 3 mice. Now, looking at a tumor growth when they're co-injected with their tumors, the tumor cell line. And as you can see here, without their presence of induced T cells that recognize the OT1, that contain the OT1 TCRs, you can see very rapid tumor growth. However, this is highly suppressed when they're induced T cells expressing the OT1 TCR when they're present. They further went to look at the tumors. Here, tumor 1, tumor 2. They looked at the presence for their TCR alpha beta induced TCRs. They then went on to look at C62 ligand by CD34. You can see that there's active T cells as well as memory T cells present. And the CD8 is also expressed interferon gamma, showing that they're functional. So that really brings me to the the last section that I want to present, and that is regarding cancer immunotherapy. Often T cells are kind of set to look like basically to give the tumor cell a kiss of death. So they interact with a tumor cell, and basically it's a kiss goodbye. When they make contact with the surface, they release basically granules that will kill the tumor. So cancer immunotherapy. This paper by Nagano uses T cells. They're specific to tumor antigen, and from these T cells, they generate iPSCs to then be able to kill the tumor. So their initial method was the following. They used peripheral blood to look at T cells that their TCR can bind MART1 tetramer. So you can see by this percentage, it's actually fairly high in terms of naive T cells, in terms of antigen recognition. So MART1 is one of the better recognized antigens for TCRs. So you see here 0.14% for CD8 CTLs. And they then take these cells and prime them with peptide to expand them. 14 days later, they prime with incubate them with peptide, and this lymphoblastoid cell line, or this serves as an antigen presenting cell. And they do the stimulation every week or once a week. And at the end, day 26, they harvest these cells, and this is what it looks like here. They look at the CD8s by MART1 tetramer staining again, and you can see that there's a high enrichment in each of these different conditions or different wells of MART1 tetramer staining. So what they then do next with these cells is to sort them allele-clonally, reprogram them to iPSCs using the Yamanaka factors. Each of these iPSCs now has still a pre-rearranged TCR locus from their T cell counterpart. And these colonies or iPSC colonies can then be cloned. And then you have clones that have specific alpha beta chain rearrangement. These can be differentiated in vitro using OP9 delta like 1 differentiation system. And then they test basically the TCR, they sequence it, and they looked at their different wells and different clones originating from different wells, and they found different VJ segments for the TCR alpha, as well as VDJ for TCR beta. So from these, they picked these clones here to analyze further. So they did again CD3 by MART1 tetramer staining, and you can see that there's some differences in terms of their affinity to MART1 tetramer, and that's also outlined here in the graph. And you see that clone 210 has a very high affinity to tetramer, and there's different affinities versus the 4-1, which is one of the lower affinities. A little bit more further, they looked at the tetramer mean fluorescent intensity staining versus CD3 mean fluorescent intensity staining, and the ratio gives again the highest sort of affinity for clone 210, and a moderate one for 5-7, and fairly lower ones for some of the other ones. So out of these, they picked. First I should mention also that they analyzed these clones for their ability to generate T cells. And you can already see that there's some difference in terms of number of cells that are here. They quantified this in terms of percentage of CD4, CD8 double positives. And clone 410 is actually very good at generating T cells. And again, there's a variability within the different clones and able to generate T cells. As well as they looked at total cell yield, and you see again differences in their potential to generate T cells, which is not surprising, but it's something that they used to characterize and pick their clones. So they picked these 3 clones for further analysis: clone 210, 5-7, and 5-10. And as you can see at the bottom here, that basically gives you the best comparison in terms of their affinity. So 210 has the highest affinity, 5-7 has a moderate affinity, and clone 510 has sort of a low affinity for MART1 peptide. They then did some in vitro killing assays using the peptide pulse T2 target cells, and they actually see that regardless whether the TCR has a high affinity, moderate, or low affinity, the killing is of the T2 target cells is virtually identical. And contrast here is a TCR specificity that's not present in the T2 target cells, so there's no killing occurring here. It's negative control. Similarly here for the melanoma MEL624 cell line, again, we see virtually identical killing, high, moderate, and low affinity TCRs, and no killing for negative control. So that leaves them with the following sort of scheme of regenerated CTLs and looking at their alloreactivity. So you've got your donor that you can get your T cells from and then generate T iPSCs with the tumor specificity. Then you can generate CTLs, and the CTLs, if you remember, they're stimulated by these lymphoblastoid cell lines, or LCLs. Of course, you can then, so this is a mix here of the LCLs with the CTLs. And what you then have is, if you pulse them with peptide, you have activated T cells. However, there's also some potential for background or alloreactivity in the system using the same donor and donor CTLs and LCLs. So what this looks like here, CD107A by CD8, you can see there's no stimulation here, or no stimulator, so no LCLs, no peptide. This would be no stimulation, but with the same LCLs from the donor as the CTLs are. And it's a very low background staining or background expression of CD107. Now, when you have the peptide present, of course, there's activation, so it's just comes to the positive control. So I want to point out again, this would be the background, so low reactivity. Now, they looked at the different HLA molecule expression of the donors, so the CTLs, and they found these different HLAs expressed. And they looked at different recipients now at their LCLs and their HLAs. So what they did here is they basically put a box around the HLA molecules expressed in the recipient that's also expressed in the CTLs. And you can see there's a very wide differences of HLA expression, and some of the recipients do not express any of the HLAs at all. So basically, this is the CD107 expression again, and you can see that with the high, moderate, and low TCR affinities, you have, in most cases, no alloreactivity. Some alloreactivity for the moderate, I would say, in some of the recipients for this one cell line. So overall, this is just very exciting news that they can use one donor CTL, and this can go into many different recipients with low or no alloreactivity. So that concludes the third paper and brings me to my summary. So basically, I hope I showed you some evidence that in vitro T-cell differentiation can be used for developing therapeutic strategies and modeling diseases in vitro, and that PSCs, T-cells derived from PSCs can be a very powerful therapeutic tool that has clinical applications if further developed. And also off-the-shelf cell therapeutics can be produced that have low alloreactivity, and they can even be further modified in the future to eliminate any sort of chance for alloreactivity. And there's many recent and future advances that is going to bring that to the clinic even more so. So with that, I just wanted to see if there's any questions that you have. I'd be happy to answer any. So thank you for your attention.